It's great to be back in Toronto, and it's great to see an old friend, Hugh Wilson, who I've known since, I think, 1990. I was working out when we first met. My talk is about an hour is what I was told the budget, so nobody can pay attention that long. So ask questions, it'll break up the flow of things, and then you'll get more out of it. I always sat in the front as a grad student and would ask questions. And that's something I encourage the students to do, especially. The faculty can ask questions if they really have to. Okay, so this is a talk about the relationship between information theory and biological systems, or information theory as a tool for understanding biological systems. And for those who aren't familiar, the fundamental construct in information theory is a channel, a communications channel. So you have an ensemble of inputs of some kind, an ensemble of outputs. The outputs are corrupted or influenced by some sort of noise along the way. This is a diagram from Shannon's classic paper introducing the whole setup back in 1948. And the thing that one typically wants to do is calculate the mutual information between the input signal X and the output Y. That is to say, if you don't observe the output, you have a certain level of uncertainty about the input. Once you've observed the output, you have less uncertainty. And the reduction in uncertainty is something that you can measure using a quantity like bits or bits per second. And that's the mutual information. And then if you try to be clever about the way you use the channel, you might find a way of packaging the information or packaging the inputs that maximizes the mutual information, that reduces the uncertainty to the greatest extent possible. And that would be called the capacity of the channel. So there's a tremendous amount of work defining different kinds of channels and calculating their capacities. That's sort of part of the bread and butter work of information theorists. So throughout biology, we see examples of things that look like communication, and it's very tempting, and many people have dived into this over the years to try and take the framework that Shannon introduced originally in more of an engineering context. I think the original motivation was how much information can you send down a telegraph pole, a telegraph wire, to try and use this same construct to understand biological systems. And typically you're looking for what are the fundamental theoretical limitations on how a particular signaling system in biology could perform. There have been a large number of success stories in applying information theory in biology. A lot of them, or at least the ones that I'm the most familiar with, involve quantifying the performance of different kinds of sensory systems. So this is a figure from a paper by, an old paper by my postdoc advisor, Terry Sinofsky, with Tony Bell, used to be my office mate at the Salk Institute. What they did was they took an ensemble of photographs taken out in nature, forest scenes, birds in nests, and I don't know what all they had. Ensemble X. And then what you're looking to do is find the most efficient representation of that ensemble using a set of filters that you optimize using sparse representation principles. And the optimal filters end up, most of them have this character, if you look at these little squares here, where you've got sort of localized information along a particular preferred direction. So little edge detectors, if you will. And these look remarkably like the receptive fields recorded physiologically from cells called simple cells in the primary visual cortex of mammals. So if you do an experiment where you expose a cat or a monkey to different kinds of visual stimuli, you can map out the kinds of stimuli that drive an individual cell the most strongly, and those stimuli look a lot like what you would get if you applied a little filter like this to the input. And so this was seen as evidence that a fundamental way to understand what's happening in visual processing is redundancy reduction or some sort of information theoretic optimization. So similar work was done in a variety of different areas. So Smith and Lewicki, for instance, looked at, they recorded auditory or natural recordings, just like walking around in the forest with a tape recorder. And they did a similar kind of analysis. And then the optimal filters that they obtained looked a lot like the filters that you obtain by measuring individual cells in auditory cortex. So once again, the fundamental processing units in the brain seem to be consistent with an information theoretic description. Other kinds of applications, in this example down here, you can see my cursor, right? Good. Okay, so this is from a paper by Vergosola et al. Called introducing the notion of infotaxis. The idea is that there's a female moth sitting here secreting pheromones that are blown downstream in a sort of a plume, and then the male moth is trying to find the female moth, and what moths are known to do in nature is they make casting paths where they'll fly zigzag across the direction, perpendicular to the direction of the wind, and the concentrations of pheromones are so small that they're typically just picking up one molecule at a time. So it's a very challenging signal detection and source localization problem. And in this paper, they compared two different ways, two different strategies for finding the source that you're looking for. In one strategy, you do something that's analogous to chemotaxis, which I'll say more about later, where you basically are trying to maximize the expected value of the concentration of the signal. So you're trying to move up the gradient. You're trying to estimate what the gradient is and move up the gradient. The other strategy was based on maximizing the information that you have. So forget about the gradient itself. I just want to reduce my uncertainty about the source, as opposed to maximizing where I am in the concentration field. You might think that these were equivalent, or you might think that, of course, what you're trying to do is get to the source, so you should climb up the concentration gradient. But what they were able to show, both analytically and through some very sophisticated fluid dynamic simulations, was that actually following the infotaxis procedure got you to the source faster on average. So for those who know this lingo, the mean first passage time from the start to the ending point was shorter if you follow the infotaxis, if you try to maximize information rather than maximize just the signal itself. So seen as another success story for using information theory as a framework for understanding a biological system. One more example. So this is work by my colleague Andreychenko, that appeared in the journal Science. They were able to measure quantitatively the channel capacity of a particular biochemical signal transduction pathway by applying a range of different levels of inputs to a population of cells, and then measuring the individual responses of the cells. So if you have one cell, you give it a chemical signal, it responds by secreting some other signal or by activating another pathway. And if you can control the input and measure the output at the level of individual cells, you see that there's a distribution of outputs for each input. And so then these are very data-intensive, high-throughput experiments where they had to be able to measure all the responses of each cell individually, and once they were able to do that, they could construct the conditional probability of an output given an input, and that is what defines the information channel. Given the input, what is the distribution of the possible outputs? And they were able to show that this particular pathway communicates somewhere in the neighborhood of 1.1 bits of information per use of the pathway, which I think some people were surprised that it was so small, considering all the effort that they went to measure it. Okay, I'll leave that discussion for another time. The point is they were able to come up with a fairly precise quantitative measurement of the channel capacity in Shannon's sense for a specific signaling pathway. All right, so motivated by these success stories and by other considerations, a whole raft of people over the years have argued that information theory should be seen as a central explanatory principle in biology. And here's just a couple representative quotes. Christoph Adami, who is both a very talented theoretical physicist and also one of the leading authorities on the origins of life and on modeling artificial life, has asserted that once we discovered DNA and the genetic code, that made clear to everyone that information is the central pillar in any attempt to understand life. Daniel Polanyi, who studies perception action loops and builds models of robots navigating the world, and sort of how would you organize agents to take best advantage of the world around them. He's made this argument as clearly as I've seen it made anywhere. If organisms would develop a suboptimal information processing strategy, that is to say, if you built an organism or designed an algorithm that did not make the most efficient use of information possible, then you would obviously be wasting metabolic energy, and such a strategy would be selected against by evolution. Okay, so there's a presumption that you find by authorities in the field that in order to get the most that you can out of life, say to maximize your fitness if you're an organism, you are necessarily going to have to be maximizing the information that you get from the environment. Ilya Neimanman, a biophysicist for whom I have a tremendous amount of respect, he wrote a paper whose title says it all. Life is information processing. Right? That's the whole story. Andrew and I were at a workshop in Banff International Research Center just last summer, and Andrei Levchenko, who I mentioned before, was repeating this same line that biological processes have evolved to use matter to generate, preserve, and transmit information. So there's a line of thought suggesting that information is pretty much the whole story. Now, that's the introduction to my talk. The first part of the talk, I will go more in detail into some of the work that I've done using information theory as a framework for looking at cell signaling specifically. And a lot of that work is joint work with Professor Eckford. So if you're one of his students, you probably know this story already, and I apologize if you're feeling like you've seen it all. You can get up and stretch, walk around the room, I won't mind. Everyone else should pay attention, because it's really cool work that Andrew played a big role in. The second half, I will change gears, and I will offer a critique of information as an explanatory principle, that is to say, by itself. From my point of view, it's maybe half of the story, and there's a fundamental piece that needs to be taken into account to complement it, and I will discuss that in the second half of the talk. So before I dive into the next bit, are there any questions? Anybody's just puzzled about something, or comments, or there was a whole flock of students who came in at the last minute, and I guess you guys are in the right place? Yeah? Okay, that's fine. Just want to make sure. Any questions before I go on? All right. So I mentioned before signal transduction pathways. So cells get information about their environments by monitoring. We could turn the gain down a little bit more. I don't know if that's under your control or if I can control that. I feel like I should be standing in exactly the right spot, otherwise something bad happens. All right, so the whole metaphor of signal transduction assumes that there is some sort of environmental signal, or maybe a signal in one part of a cell that the other part of the cell needs to know about. And there is a noisy process by which the information about the outside world is conveyed to the recipient. Now, in order to do a quantitative study, you have to really get the noise right, because the noise is what defines the channel. And then you have to spend a lot of time, if you're working in this field, understanding the biophysics of what's actually happening in the signaling process. Here's some nice examples. This is a classic movie here of a Dictyostelium discoideum amoeba. These guys, they live as single cells out in the wild until they run out of food. When they start to starve, they aggregate into a multicellular organism, and the way they do that is by secreting cyclic AMP to signal to each other and then crawling towards the local source of cyclic AMP. What you see here is a pipette with cyclic AMP in it, and this poor amoeba is like chasing it, doing chemotaxis, so it's moving up the gradient, and there's actually a bunch of amoebas around, and you see some of the other ones are getting in on the action too, crawling in from off screen. Okay, so this is a classic example of a chemotaxis process where the cells are detecting a gradient and then moving towards it. So I looked at this process a number of years ago, and actually before I started working with Andrew, and said, well, it looks to me like the cells are processing information. There's a signal, they're getting it, the signal into the cell by the binding of cyclic AMP to the receptors. So here's an abstract model of a cell. We've turned this, instead of a spherical cow, I have a spherical amoeba here. So my spherical amoeba is studded on the outside with maybe 10,000 receptors. Each is can have two states, either bound to a molecule of cyclic AMP or not. The little blue dots, if you can see them, I think you can see them, these are individual molecules of cyclic AMP that we simulated. They're doing a Brownian motion in this little volume here, and they're bouncing around, and when they bounce into the cell, they have a probability of sticking to a receptor. Now, the cell's job is to take, it can't see the gradient. All it can see is what its receptors are doing. So imagine yourself sitting in the center of, I don't know, the Astrodome or a planetarium or some space where there's lights all around you, and they're flickering on and off. So every time there's a receptor bound, the light bulb is on. Every time it's unbound, it's off. And there's a very slight bias where the turning on is happening a little bit more on one side than the other, and your job is to look around and figure out which direction the bias is in. That's the job that the cell is doing, trying to make a decision about which way the gradient is based on its receptors. And so we built a computational model in which we applied a small gradient. Here you see there's a gradient, hopefully you can tell. There's more molecules on this side, so there's a source on this wall, and on this wall it's absorbing, so the concentration right up at the wall is 0. So now you've got a nice linear gradient from one end to the other, and so you'll have slightly more bound receptors on one side than on the other side. And if you kind of just take each receptor weighted with a unit vector, you take a weighted sum, you get a little vector that gives you an estimate of what direction the gradient was. A priori, the gradient could be in any direction. And if you're doing an information theory calculation, you need to know what the distribution of the signal is before you observe it and after you observe it. And then comparing the entropy of those two things, you end up with the mutual information. So we did that calculation, and you can see here in the shallow gradient, there's a slight shift in these vectors which represent the estimates. So initially they're distributed in every direction, and then when you turn on the gradient, they shift over to one side. But it's pretty noisy. The cells, the reduction in uncertainty is not that large. If we make a steeper gradient, then when the signal turns on, there's much less uncertainty. Okay, the estimate is much tighter. So if you think of the distribution across the cell of the posterior distribution, the entropy has dropped significantly. And so here is where we calculated the entropy in bits as a function of time after you've turned on the signal. So initially, this value up here, between 3 and 4, that's just the natural log of the surface area of the sphere, right? That's the entropy of the uniform distribution on the sphere. When you turn on the signal, it drops, but then eventually, if the concentration is too high, the receptors saturate, and then you sort of become blinded again. So there's actually an optimal time to make a decision. You have more information after 2 seconds than you do after like 5 seconds. Okay, so I think for the sake of time, I will jump ahead a little bit. So there's other people who have used information theory and chemotaxis as well. I'll skip over that. All right, so another example that's related is supposing that we just, instead of trying to detect a gradient, suppose we're just trying to detect a fluctuating signal. So in this illustration, we've got a cell here that's secreting molecules. Think of them as cyclic AMP molecules if you like. This cell is counting the number of molecules either that bounce into it or that are in a close proximity to it. And so if this cell is putting out molecules faster, slower, like an oscillating signal, then the concentration of or the number of molecules that are close enough to bind to the receiver, you can see is also going up and down in a kind of sort of sinusoidal way. But then if you look at an individual receiver, which is either bound or unbound, what you see is this up and down. And this is the signal that the cell would actually see. Excuse me. And so you can begin to see what a challenging problem the cell is facing, right? This is the intended signal, or the signal as sent by the sender, but then all you see is this sort of flicker. And when the concentration is high, the flickering is happening faster, and when the concentration is low, the flickering is happening slower. But so you've lost a lot of information, introduced a lot of noise, and that's the sort of thing that we're trying to understand. So this is a case where you just have two states for the receiver. It's just bound or unbound. But in other systems, you can have more complicated state diagrams for the process that's receiving the signal. So for example, there's a molecule involved in synaptic plasticity called calmodulin, which binds calcium. It's a second messenger involved in calcium signaling, and it's got maybe 8 different states. And some of the transitions between the different states are driven by the calcium concentration. Some of them are not driven by the calcium concentration. And so you can say, well, what's the mutual information between the calcium signal, if it was fluctuating up and down, and the state of this receptor? And it can get even more complicated. Calmodulin has got tens of thousands of different states, for instance. Okay, so this is the point where I started doing work with Professor Eckford, and we looked at the simplest case of this kind of interaction between an incoming signal and a signal transduction process that involved binding and releasing the signaling molecules, and having a receptor that has different states, and you're transitioning between the states in a way that depends on the external signal. So here's the external signal concentration. There's a receptor on the cell that has 2 possible configurations, either bound. So here we've bound to one of these ligand molecules. Ligand is just a word for stuff that floats around and binds to something. So that's the signal. And then the unbound state. So what's different in this system than in maybe more traditional engineered information theoretic devices is that only one of these transitions is sensitive to the signal. When you're unbound, the rate at which you become bound is sensitive to the signal, but when you're bound, the rate at which you release the molecule and go back into being unbound is insensitive to the signal. So it's sort of like a receptor with dead time, if you've studied those kinds of things. So the probability of being in the bound state just follows this simple example of what we used to call the master equation, but that's not good language anymore, so we call it the forward Kolmogorov equation, which is a big mouthful. P is the probability of being bound, and so 1 minus P is the probability of being unbound, and so this gives you the rate at which you become bound if you weren't already, and C is the concentration signal. So this term would be alpha, and then K minus is the same thing as beta, that's the rate at which you're going back. So we analyzed this system, and we were able to obtain the mutual information and the channel capacity. To do that, we looked at it in a discrete time approximation, and long story short, by leveraging previous work by other people in information theory, including Toby Berger, who's a big name if you're in information theory, we were able to find the mutual information, and by maximizing that, we were able to find the capacity. And we were also able to show that the capacity can't be increased by feedback. What that means is if the receiver somehow, if the sender is somehow paying attention to what message the receiver got, in some cases you can adjust the way you send the signal based on whether it was received properly, and you can kind of game the system to improve the channel capacity, but that doesn't actually occur in this system. Okay. There's a classic paper by a Russian probabilist named Kabanov where he looked at an even simpler system where it's called a Poisson channel, where I just have a flashlight and I'm turning my flashlight on and off or dialing up and down the intensity of my flashlight and trying to send messages that way. The statistics of the photons that you receive are Poisson distributed, and they're intrinsically variable or noisy. And by doing this really beautiful information theoretic calculation, he's able to derive this horrible looking formula for the capacity of a Poisson channel where lambda and m are related to the maximum rate at which you can send the signal at the maximum intensity of your flashlight and the minimum intensity. If you can shut it all the way off to 0, then the capacity is infinite. So he has to assume that there's, even if the flashlight is turned off, you know, cosmic rays or something are giving you a little bit of background noise, okay, the dark current. So it turns out in our binding, unbinding system, if you make the unbinding super fast, so if whenever a cell gets bound, it immediately releases, and then so it's never in the blind state. Then all you're doing is counting the concentration, which is like a Poisson signal, because the binding process is just well described by a Poisson random variable. And so it was very satisfying. We were able to take these somewhat complicated expressions that we got, and by doing a little bit of calculus and taking limits, we were able to get these other really complicated expressions. But they were ones that agreed with what Kabanov had done, 40 years earlier or something. Okay. Let me skip to... okay, one more piece on this, and then I'll change to the second part of the talk, because I do want to have time for that. Okay, so in a further elaboration, building on that work, we've looked at situations where the receiver is not just a two-state process, bound or unbound, but where the receiver has multiple states, which is trying to make things a little bit more biologically realistic. One example is the channel rhodopsin molecule. So this is a molecule that's become hugely important in experimental neuroscience. It's the basis of optogenetics. So you can have these ion channels that, when you shine the right kind of light on them, you can manipulate cells in neural networks, biological neural networks, in unprecedented ways. The simplest model for channel rhodopsin has two states, a closed state, two closed states, and an open state. So when the state is open, that allows a current to flow through the cell, and then that current can be detected either experimentally or else it affects the electrical behavior of the cell. When you're in the closed states, that current is shut off, and the received signal is effectively zero. Now there's these two different closed states, and you can't tell which one you're in if you're not seeing any signal. Okay, so the underlying structure is a three-state Markov process with, in channel rhodopsin, this transition from the from closed state number 1 here to the open state, that's driven by light. So it's kind of like the Poisson channel, where I have my flashlight, and when the intensity is higher, it's driving you across this transition. Then this transition is not affected by light, and this one is also not affected. But the new wrinkle here is that you got these two different states that you can't tell them apart. So you've got a three-state Markov process, but what you observe is just a two-state process, either open or closed, and it's no longer Markovian. For those who have worked in information theory at all, you'll know the distinction between a process whose future can be predicted based on the present state just as well as it can be based on the entire past. Okay, so a process that is conditionally independent of the past given the present, and everything else, which is not Markovian, where knowing the full history gives you additional information about the future. And if you're just observing the projection onto open versus closed, you no longer have a Markovian system. It's a heck of a lot harder to analyze, also more realistic biologically. And so we've put a fair amount of effort into trying to understand these systems in a similar way to the level at which we understand the simple binding, unbinding system. There's lots of examples of these things. I call them partially observed Markov processes, where the transitions are driven by the intensity of the input. So there's a molecule involved in the control of the motor system. It's part of what's keeping us all alive and moving, called acetylcholine, and it binds the acetylcholine receptor, and that has 5 states like this. 2 of them, the channel is open, 3 of them, the channel are closed. These bold arrows are driven by the binding, so they're sensitive to the concentration of acetylcholine, and the smaller arrows are not. And here's calmodulin again, which I mentioned before. So all of these have this sort of structure where you have some edges driven by the input signal, and some of the transitions can be observed and some of them can't. And what we find is that you can calculate the mutual information between the input and the output if you could observe the full thing, because then what you're looking at has this property of being Markovian. Numerically, you can evaluate the information you gain about the input if you just look at open versus closed. So if you lump these states together, and in this case, you get a huge gap between the information. So if you can only observe open versus closed instead of all 5 states individually, you lose like 90% of the information. In this case, with channel rhodopsin, you don't lose very much of it at all. You have almost the same information about the light source, regardless of whether you can distinguish all 3 states or if you can only distinguish these closed versus open. And so we have an ongoing project where we are trying to understand exactly, what can we say about where that gap comes from. Okay, so that's the end of the that part. Oh yes, question, go ahead.
So before we look at the more complex systems, can we go back to another slide?
To the 2-state process?
Yes, that's
Okay.
2-state. So I understood that if that beta was 0 and it was on down very fast, the acquisi there would give us direct information about the density of the ligands, and we can totally predict that. But I don't even have a slight idea what would you do if beta wasn't 0.
How to analyze, so the question is, how do you analyze the system if beta is finite? Yeah, so the case that is trivial, well, it's not trivial to analyze, but it reduces to Kabanov, which was done before, is when beta goes to infinity. If beta is 0, then I just sit here forever and there's no information. But when beta is finite, so what we do is first of all, we go to discrete time instead of continuous time, because doing rigorous analysis of this kind of process is more difficult in continuous time than it is in discrete time. So now we have discrete time steps, and we've got 2 possible states for the output, and for the input, you have to make a choice about what kind of ensemble you're going to put into the system. So we decided to have the input concentration over some range. So there's a maximum concentration and there's a minimum concentration. And we thought about, on each time step, let's draw from some probability distribution of the possible inputs. Well, it turns out that if you're trying to maximize the mutual information to find the capacity, if you want to get the best use of the channel, it's a waste of resources if you use anything except the highest and the lowest concentrations. So the most effective way to use this channel theoretically, I'm not saying this is how it works biologically, but to do the theoretical calculation, you flicker back and forth between the highest and lowest concentrations. Now, you could do that so it's 50-50 chance of doing each, but you could also do like 80% high and 20% low, and there's a trade-off, because if you spend a lot of time high, then this will spend a lot of time bound, and when it's bound, it's insensitive. So there's a trade-off, there's 2 different effects. And that's why did I show a plot? Yes, I didn't actually show the plot. If you vary the probability of sending high versus low, the mutual information goes up and comes back down. That's finding the maximum of this with respect to that parameter. So, so then these calculations, this is the entropy function, P log P, the sum over the possible, either being bound or unbound. And this is the conditioned entropy and the unconditioned entropy. So this is the uncertainty if you haven't made the observation of the output. This is the uncertainty if you have. And this down here is factoring the fraction of time that you're in the sensitive state. When you're in the insensitive state, the mutual information is 0. So you take the information you would get if you were in the sensitive state, and you multiply that by the fraction of time you're in the sensitive state. And everything converges to that when you do it all formally. Okay? Does that help?
It helps. Yes,
And I'm happy to, we'll have lunch together with the students, and there will be a whiteboard, and I can talk more about that then. Yes?
In your case where you have non-Markovian,
Mm-hmm.
how do you bring in the signal that turns you the unknown transitions? Do you have another noise there that does that?
Right. So we can think of this variable y as the state of the channel. So the fully resolved 3 states here or 5 states here. Z would be the projection down to just binary open versus closed. And then X, which is the input, we need some model for X. So once again, in this case, it's driven by light. So light intensity could be any function you like. It turns out the best way to use the channel is to flicker between all maximum light and minimum light. Okay, so you're going back and forth as either as a white noise process with these 2 different values where you bias how much time you spend high versus low, and then that parameter is something that you look at things as you vary that parameter. Or we've also been looking at the input being, say, a Markov process itself. A 2-state Markov process where it just goes up and down, or you could imagine a population of sending signals that are sending inputs that are obeying some sort of a Markov process. As a physical example, you might think about the dendritic connection between 2 nerve cells, where action potentials are coming from the first cell. They cause the release of neurotransmitter that diffuses in the synaptic cleft. So in that case, you might have like a Poisson process input, and then you might have sort of like little exponentially decaying bursts of neurotransmitter. So then your input signal X would have some more detailed structure to it.
Is the 2-state closed one sort of like bound and unbound, where if you get bound, then it takes a random time to get unbound?
That's yeah, that's right. So once you're here, you're insensitive. Right, you're insensitive if you're in state C3. You're also insensitive if you're in state 2. When you make this transition, you know you've made that transition. So as soon as it's happened, you know you're in state C3, and then there's an unknown length of time before you make this. So this is close to that binding-unbinding channel. And qualitatively, this transition is both observable and sensitive to the input, which is why it doesn't really make any difference whether you can observe the full channel or not. Whereas in this case, the transitions that are directly affected by the input are not the ones that you can observe. So that qualitative description explains why there's a gap. We're trying to make it quantitative. Yeah. Good. Other questions before I go into the last part of the talk? Okay, so I brought this slide back up that I showed before about a variety of prominent authors who have claimed that information theory is the key to understanding biology. And for the sake of a straw man, I'll call it the strong hypothesis, that behaviors that are optimal for an organism's survival are necessarily information optimal. And the reason I'm putting this out as a straw man is because I'm going to give you 2 counterexamples that show that this is not the case, and that there's more to the story. Now, what's the part that's missing? You can think of it as the presence of constraints that occur naturally in biological systems, or the need to take into account both information and energy, if you like. If you are familiar with thermodynamics, the free energy is a term that involves the energy and the entropy, both. And if you really want to understand what's happening, say, in a chemical reaction, you can't just look at the entropy or just look at the energy. You have to take H minus TS and take them both into account. So in a hand-waving kind of way, that's sort of what I have in mind in the background. But it's what we've done is construct analytically solvable models where you can look at this question. And I think it's useful to have simple models that can be analytically studied. Okay, so the general framework here is a perception-action loop where you've got stuff happening in the world. Maybe the world is represented by a 2-state Markov process. You're observing the world. You're taking your observations running them through some sort of strategy and making an action that then changes the world. If you've seen Markov decision processes, that's a framework that's similar to this. So here's the first example this is work I did a few years ago with a colleague, Hillel Cheel at Case Western. We've simplified things a lot. And what we're doing is we're imagining that there's a creature living in a one-dimensional world. So it's on like a little railroad track that goes around and around. And the creature is trying to find a source of food. So S here is the source. And this is loosely, well, it's inspired by Vergasola's infotaxis work, but instead of having a moth releasing a pheromone plume, we've just made the world one-dimensional and finite. And the source is releasing some sort of diffusible chemical, and the creature can detect the local concentration of that chemical. And if you've simplified things enough, basically what the creature can do is it says, well, if I know everything about the physics of the world, if I know what the concentration is, I know how far away I am from the source, right? Given the level of concentration that I observe, I could be, suppose I know I'm 1, 2, 3, 4, 5, 6 ticks away from the source. I could be here, or I could be down here. I don't know which way the source is from me, but I know how far away I am. And then we let the creature move wherever it wants to move. We invent different creatures with different strategies. So we have an infotaxis kind of strategy, and we have a chemotaxis kind of strategy, and we have some other strategies. And then for each strategy, it leads to a Markov process where we can calculate the stationary distributions, and we can calculate how much information the cell has about the source, and also how much food it gets, and we can just compare these different quantities. Now, in distinction to what Vergasola was doing, our source is moving around. So if the female moth is like wandering around, that's a different system than what he was doing. The moth was sitting still and waiting for maybe this is how real moths behave. They wait for the mate to come to them. There's more one could say there, but I won't since I'm being recorded. But in this case, the source, the target, is moving around unpredictably, okay? So not totally unpredictably, it's just doing a random walk where it either moves right or left with probability x and stays where it is with probability 1 minus 2x, and then we can vary x over some range. Okay, so what does the creature do? Well, once the creature has made an observation, we can quantify what it knows about the source. In some cases, like if it happened to be so lucky as to sit on top of the source and the distance was 0, then it knows where the source is. Or if it was on the other side of the world, or maybe by looking at a series of observations, maybe it would know where the source was exactly. Or it could happen to know, it could be in a state where it knows that the source is either one to the right or one to the left of a definite position. So there's these different states, we call them information states, after you've made an observation. Now, knowing where the source was, or where it could have been on the last step, doesn't tell you where it's going to be on the next step because it takes a random step. So, but if we convolve each of these with the distribution of the next step, then we get the possible distributions for where the source is one step later. And so if I knew where it was before, then this is the distribution, the posterior distribution for where it's going to be on the next step. And this is if it was 1 away, then the next step looks like this. If it was 2 away from a definite place, it looks like this. If it was more than 2 away, it looks like this, etc. Okay, so you've got these different a posteriori distributions that capture the knowledge that the creature has about the source. So now we can formulate different strategies for what should the creature do once it is in one of these information states. Hugh.
Peter, I think I'm missing one thing, but if the substance diffuses in both directions, wouldn't the creature have a high probability of sensing that it's higher concentration in the shorter distance than in the longer distance?
It doesn't know where the source is. So if I hear your voice and I know that you're, 5 meters away from me, but I don't know if you're over here or you're over there, that's the situation the creature is in. So if I go back to the original picture here.
Being on a circle, why
So the
substance diffuse the other direction?
It does. So if the creature detects that the source is, what did I say, 6 units away, then 2, 3, 4, 5, 6, then the source could also be down here. The source, given that information, I don't know whether the source is at this location or this location.
So the creature does not have 2 sensors, one.
Yeah, so if I get there, the second example, the creature is doing gradient sensing, but not in this case, it's just doing, that's right, it's just doing absolute distance sensing. We simplified a lot, but in exchange for that, we got an exactly solvable model. Okay, so now we have these different strategies. First of all, the infomax strategy. How should the creature move in such a way that it always maximizes the information or minimizes the uncertainty? Another thing we call the maximum likelihood strategy. How should the creature move so that it ends up at the most likely place? So it maximizes its probability of encountering the food source. And another strategy sort of switches back and forth between the previous 2 strategies depending on which state it's in. So, for example, if I'm in this state where I know exactly where the source was on the last time step, and this is the distribution of where it is on the next time step, in the maximum, the likelihood maximizing strategy, I'm going to move to the peak of the distribution, right? So I'm going to go to the most likely place for it to be. But on the next move, it could move right, or it could move left, or it could not move. And if it moves right or left, now I know I'm 1 away from it, but I don't know which way it is. Okay, so I can maximize the chance of hitting it at the cost of losing information. If I want to maximize information, if I, instead of landing on top of it, I choose this red location and I move 1 next to where it was last, then I'm guaranteed on the next step, either I hit it, or it's 1 to the right, or 2 to the right. But either way, I know exactly where it is. And so in this simple model, by never landing directly on top of the source, I always know where it is, but I never get fed. It's like a lion hunting a pack of gazelles, and it stays in the grass and it never pounces. Once it pounces, they scatter, it doesn't know where they are. So if its motivation is to maximize information, it can do that perfectly happily while starving to death. And the point is just to illustrate that maximizing information doesn't have to be equivalent to maximizing your utility or your survival or your reproductive success, right? That's the point of this simple model. Okay. Now this clock says there's 5 minutes till the hour, and that clock says it's already, do I still have 5 minutes? Okay, so I will skip the analysis, but we can solve, yeah.
Started 5 minutes after, so if you...
Oh, if there's a little wiggle room, okay. So for each of those 3 strategies, we end up being able to, we have an exact representation of the model as a Markov process on a finite number of these so-called information states. And so for each of those states, we can calculate exactly what the mutual information is, how much uncertainty the creature has about the source, and we can also calculate how much food it gets. If we make the food it gets, say, a function of how far away it is. And so, not surprisingly, as we vary this parameter here, this is the source mobility. So this is how random the movement is, right versus left. The information theory optimizing creature always has better information than the other ones. The one that is greedy has the worse information. I mean, not dramatically worse, but consistently worse. And the hybrid one, which switches between the strategies, is in between. So far, so good. But the next plot shows what the expected amount of food or the expected utility is for different levels of source variability for the 3 strategies. And what this shows is that there's no pattern. There's no rhyme or reason. There's no consistent relationship between being better at getting information and being better at getting the food. So the information theory creature turns out to have the greatest success in getting food in the case where the source is moving the most randomly, which is suggestive, but, so far it's just an interesting observation. The greedy creature, the one that jumps to the peak of the probability distribution every time, is this blue curve. It's the best strategy for intermediate ranges. And then the hybrid creature, which switches back and forth between the strategies, It's the best of the strategies when the source is the most predictable. So the conclusion is there just isn't any simple relationship between maximizing information and maximizing how much you get fed. Okay, so I think there's just enough time for me to squeeze in the second counterexample, which I will do here. So this is similar to the previous model. There's a ring, there's a creature moving around a ring. This time the creature can detect gradients though. And it's easier if I just show you the picture. The main difference is now we've got two different kinds of food. The creature is eating substance A and substance B. A and B are both distributed spatially around this ring. It can detect local gradients using receptors. So it's got receptors for A, receptors for B. It has receptors at the right end of the cell and the left end of the cell. It has an internal store of the two kinds of nutrients. And if it runs out of either, it dies. If it accumulates a sufficient level of both, it divides into two cells and life goes on. For that cell, there's an obvious measure of the fitness, which is the asymptotic growth rate, the logarithmic growth rate of the population. So how rapidly is it dividing? But you can also ask how much information does it have about the environment by calculating the mutual information between the local concentrations and then what's happening at the receptors. Now the constraint that we impose on the cells is that they only have a finite number of receptors. We give each cell 400 receptors. They can put 100 for A on the right and 100 for A on the left, 100 for B on the right, 100 for B on the left, and they can then calculate their gradients and move around. Or if they are about to die because they don't have enough of nutrient B, they might rather shift some of their receptors from type A to type B because they need the information more about one substrate versus the other. The reason we're doing this is it's a long-standing problem in information theory how you put meaning or semantics or subjectivity into a framework consistent with Shannon's original theory, which is a purely syntactic theory based only on the statistical structure of the world. But in biology, signals mean something. Every time there's a communications channel in a living organism, it's there to serve a purpose, to keep the animal alive, to keep the animal from being eaten. And if you try to take into account the task that the information is supposed to be useful for, we'd like to have a way of distinguishing why some information is more useful and other information is less useful. And as of today, nobody has done this, despite since 1948, everybody who cares about information theory and biology knowing that this was a crucial thing to do. This is still a fundamental open question, how to do this. Okay, so in our simple little model, there's these two substrates, there's information about A, there's information about B. And the thing is, if I move my receptors from A to B, the number of bits I give up about substance A is bigger than the number of bits I gain about substance B, because the entropy function is a concave sort of logarithmic function. So I give up more than I gain. So am I willing to give up 2 bits of information about something I don't care about in order to get 1 bit of information about something that is life or death? Well, obviously, yes. So, all right, so we do simulations and these creatures move around, and I'll wrap up about here. So this slide shows how the mutual information, the information that the cell has about the world at its by observing its noisy receptors, how that differs between 2 different kinds of cells. One always has 100 of each type of receptor. That's going to have the most information because of this logarithmic property of the information curves. The other cell is going to reallocate its receptors based on what it needs. So it has an internal state, how much A and B it has, and if it's running out of A, then it puts more of its receptors into type A. If it's running out of B, it puts more of its receptors into type B. So it's giving up information for the sake of its own survival. We have a parameter called the cell stress, which is basically the metabolic cost of just being alive. So how rapidly you're burning up your A and B just by moving around. And so we vary that parameter. But no matter what value we choose, the cells with the equal receptor strategy have a larger mutual information between the external world and the receptor state than the cells that are dynamically reallocating their receptors. But if you look at the growth rate of the population, the relationship is reversed. So, of course, the bigger the cell stress, the slower the population grows. But in every case, the population that has the better information about the world in total does worse than the population which is sacrificing less useful information in order to use the more useful information. Okay, so we think that this is an approach that gives us a window into defining a notion of subjectivity or information processing that is determined in part by the internal state of the organism in a way that avoids hand-waving and touchy-feely stuff, and is hopefully remains rigorous and still makes contact with the original formulation by Shannon, which has been hugely successful. So this is something that Andrew and I are exploring, and also my collaborators in Nebraska, Tyler Barker and Max Pirabon, and hopefully we'll have more to say about that in the near future. Okay, so that's the end of the talk. I know I hit a lot of things. I appreciate you all. You're all acting like you're still awake. That's really impressive. That wouldn't happen in the United States. Oh, is this being recorded? All right, sorry. So just 5 conclusions. Information theory does provide a natural framework for quantitatively analyzing biological signaling. Right? It's been used successfully in many examples. And we've used it to obtain the capacity of this binding channel, this on-off two-state channel, and it's a natural generalization of this classic channel from Kabanov. Generalizing from that, if you've got Markov processes with transitions, the intensity of the transitions is modulated by the input signal. We think that's a natural class of models for studying signal transduction generally, and a useful one. And then from the second part of the talk, optimal survival strategies are not necessarily optimal in terms of information if they're optimal for survival. So it's easy to find examples where the two simply are not equivalent, despite theorems that have been proved in the literature that claim that they are equivalent. And finally, if you're looking for a nice thesis project or a way to have an intellectual impact on the field, figure out a way to define subjective or semantic information to make information theory even more useful in biology. All right, that's it.